This conference will now be recorded.
Good evening. I'm Dr. Divya on behalf of TeenEinstein. So, in this chapter, principles of inheritance and variation, till now we have learned so many concepts about this. We learned about genetics, what is genetics, and who is the founder of that branch, or who started the first genetic experiment, or with the proof of experimental data, who tried to prove it. Mendel was the first person who studied the phenomenon of the segregation or gene expression, all these things in a proper way. Or that was the first reported systematic study in the case of genetics, right? So studying all this pattern of inheritance in pea plant, he selected 7 contrasting characters, and from the results he proposed the principles of inheritance. And now we are calling this as Mendel's law of inheritance. And he proposed that factors, he called that, nowadays we are calling as genes, but earlier Mendel was calling that as factors. And these factors, they are regulating the characters, and they are seen in pairs which is called as alleles. Okay, so he observed the expression of these characters in F1, F2 and all. In different generations he observed, and it was in a particular ratio, and he reported the particular ratio. So some characters were expressive, like some characters are dominant over some other characters. Okay, so the dominant characters, they are expressed when that factors are in homozygous condition or in heterozygous condition. In both conditions, they will express. Even one allele of that dominant character is present, means it will show the dominant trait or dominant character only. And when that recessive character will be expressed, when both are in homozygous recessive condition, then only we can see the recessive character as an expression, as an expressed character we can see only when it is present in homozygous recessive condition, right? So according to Mendel's law of segregation, these characters, they will segregate while the formation of these gametes. So gamete formation and the segregation of characters, that is explained in the law of segregation. So some characters and some other characters, they will show some incomplete appearance. That is called as incomplete dominance. And some will show along with the another gene also will get the expression. That is called as codominance. So when Mendel studied all these inheritance characters, he found that the factors, they are independently assorting and they combine in all different combinations. Right? So these different combinations of gametes, they are represented in a square format or tabular format, that is called as Punnett square. Reginald G. Punnett, he only proposed that. So the factors, these factors in chromosome, they are regulating the characters. So all these characters, they are called as the expressed one. They are called as the physical expression. That is the phenotype or phenotypic expression and the genetic condition that is called as genotype. Is it clear now? Is it on? Can you hear me?
Yes, Deepa. Sorry, yes, Divya.
Okay. Then afterwards, several scientists, they developed new concepts in genetics. So all these, the factors on chromosome, they are present on chromosome. They found out that they are present on chromosome. Then they are regulating the characters, and these are called as genotype. And the physical expression of the character, that is called as phenotype. So this is all we have learned already. And after knowing these genes are located on chromosome, they brought a new correlation between Mendel's law and the, like how they brought the segregation and the assortment of chromosome during meiosis. They studied that, how that a chromosome is getting segregated during cell division or during meiosis, and how they correlated that with Mendel's law, like that segregation. Mendel already told that law of independent assortment or the segregation, law of segregation. And they further studies of all other scientists, they try to correlate these laws, Mendel's law with the cell division and the separation of chromosome during cell division. Then Mendel's law that extended in the form of chromosomal theory of inheritance. Okay. So later it found that Mendel's law of independent assortment that like it is not holding us like true genes. Okay. It's not like that exactly how Mendel is telling. There are so many deviations from that law. Okay. Always it was not coming in the same proportion. There were so many deviations from that Mendel's proposal or the whatever Mendel proposed. So next we are going to study about, in the last class we learned about chromosomal theory of inheritance, right? So in today's class we are going to discuss about sex determination. So in sex determination, the cytological observations that made in a number of organisms, like a number of insects, that made the development of this concept, the concept of genetic or chromosomal basis of sex determination. Okay, actually this chapter is very important. You are getting so many new terms in this. Okay, so it's better to note down all these things. This chapter is very important for your NEET exam as well as board exam. So note down each and every point. So the number of insects, there are Scientists conducted so many studies, number of studies in the area of genetics, and they conducted so many experiments with number of insects, and that led to the development of another concept, the concept of genetic or chromosomal basis of sex determination. How we are going to determine the sex of an organism based on the chromosome or based on the genetic level. Okay. Henking, note down scientist name. Henking in 1891, he traced out a specific nuclear structure all through the spermatogenesis in a few insects. Okay, they conducted in insects. So how they did that study, Henking? He traced out a specific nuclear structure. A specific nuclear structure. Inside the nucleus, there is a some specific structures. He traced out throughout in his experiment. During spermatogenesis. What is spermatogenesis? Formation of sperm. So during all the spermatogenesis, he traced out a specific nuclear structure all through spermatogenesis in few insects. In some insects, he noticed this during spermatogenesis. And it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas other 50% sperm did not receive it. Okay, we got the point? While spermatogenesis, he observed some special or specific nuclear structure. It is present throughout spermatogenesis in some insects. Okay, and later they observed that 50%, not 100, 50% of the sperm. So during sperm production only or spermatogenesis of some insects only, they noticed this extranuclear structure which is present throughout the spermatogenesis, sperm formation. So they observed that this 50% of sperm during spermatogenesis, out of 100%, 50% of the sperm, they are receiving this structure after spermatogenesis. Okay, 50% are receiving after spermatogenesis, whereas other 50% sperm did not receive it. Okay, out of 100% or total sperm produced, out of that, during spermatogenesis, 50% of the sperm, they are receiving this structure, these extranuclear specific structures, they are receiving, or that sperm is receiving that particular structure after spermatogenesis, and another 50% is remaining, right? So that 50% sperm did not receive this structure. 50-50, the chance is 50. 50% is receiving that extranuclear specific structure, but 50% of sperm did not receive it. Okay. So Henking gave a name to this structure as X body, but he could not explain the significance of that. So Henking in 1891, Henking reported the specific nuclear structure. All through spermatogenesis, it is present. But even though it is present, 50% of the sperm, it is receiving this extranuclear specific structures after spermatogenesis, but another 50% is not receiving this structure. Okay. So Henking, he named this structure as X body. He gave the name as X body, but he could not explain the significance. He reported such a structure is present during spermatogenesis and 50% is going after spermatogenesis to that particular place, but 50% of the sperm did not receive, or they are not receiving that particular structure. And Henking named that structure as X body, but the problem was he couldn't find out, or he was not able to explain, or he was unable to explain the significance of that particular structure. Okay. So the geneticists, all these scientists, they found that all this genetics related experiment, it was like a puzzle in front of them. So some of the experiments carried out by insects, or by using insects, or in insects they did that experiment. And in initial clue about genetics, the chromosomal mechanism of sex determination that can be traced back. The chromosomal mechanism that can be traced back. So the cytological observation, whatever they made in the number of insects, that led to the development of, or the concept of, genetic or chromosomal basis of sex determination. Okay, so actually the cytological observation made by made by these scientists in this study of all this in particular type of insects and several types of insects, that led to the development of the concept of genetic or chromosomal basis of sex determination. Then a specific nuclear structure all through the spermatogenesis in a few insects, it was traced by whom? It was traced by Henkin. Okay, Henkin in 1891 he reported that, and it was also observed by him that, what was this observation? 50% of the sperm receiving this structure after spermatogenesis, but other 50% of sperm did not receive it. Then he named this structure as X body, and he failed, or he was unable to explain the significance of that X body. Okay, Henkin named as X body. Then how he concluded that X body, how Henkin concluded? The conclusion that X body of Henkin, it was in fact a chromosome, and that is why it was given the name X chromosome. Okay, actually that's a chromosome, and that's how they made the name X chromosome. So it was possible because of the further investigation by another scientist. All those followers, those who follow the same path of these scientists, they further made investigations and they gave the name as X chromosome. What is that? X chromosome. So further investigations by other scientists, they led to the conclusion that, how they concluded that X body of Henkin, that was actually a chromosome. X body was actually a chromosome. They gave the name as X chromosome. For that particular chromosome, they gave the name as X chromosome. So that was also observed that in a large number of insects, the mechanism of sex determination is of the type XO type. What is that XO type? XO type, that is, all eggs bear an additional X chromosome besides the other chromosome. Okay, all egg cell bear additional X chromosome besides the other chromosome. Okay, they are called as autosome. So they observed that large number of insects, mainly they used insects for their studies, the mechanism of sex determination. Then in that they found out in almost or large number of insects, the mechanism of sex determination is a exotype. What is that? What do you mean by exotype? All eggs bear an additional X chromosome besides the other chromosome. Okay, other chromosome means autosomes. Okay, so they'll be having a particular number of autosomes, all other chromosomes. Then they have an additional X chromosome beside the autosomes. Okay, and in other hand, some of the sperm bear X chromosome, whereas some do not. Okay, that was another finding. First finding was along with they were having autosome plus X chromosome. X chromosome. That was the first finding. Then some of the sperms bear X chromosome, whereas some do not. Some were having X chromosome and some were not having X chromosome. Okay. Then eggs fertilized by sperm having an X chromosome become female. Okay. Here, just see how that sex is getting determined. That eggs fertilized by sperm having an X chromosome. Okay. The sperm is having an X chromosome that becomes female, and those fertilized by sperm that not have an X chromosome become male. Okay, we said autosomes. Autosomes. These are the chromosomes which determine the other characters. Okay. So that determines other characters. So it is having, these are autosomes. These autosomes plus they have X chromosome, they said. Here, X chromosome. Okay. So when that egg here, in insects also egg plus sperm, right? There is a sperm. This is in the case of human beings, we will give structure like this. So egg plus sperm, that forms zygote, right? So in the case of insects also, this X chromosome, if it is getting fertilized by a sperm, X chromosome always it will be that egg. Here you can see that egg fertilization by a sperm that is having X chromosome that becomes female. Okay. So autosome plus already it is having an X chromosome. In some it will be having that chromosome and some that won't be having that X chromosome. Right. We have already told this. So while fertilizing, that sperm is having X chromosome, then it is forming female, and those fertilized by sperm that don't have, if the fertilizing sperm don't have an X chromosome, that becomes male. Okay, so due to the involvement of X chromosome in the determination of sex, involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome. Okay, so this X chromosome is involved in the sex determination. That's why it is called as sex chromosome. Which chromosome? That is sex chromosome. Why that? Because of the major part or the major role or the major involvement of that X chromosome in the determination of sex. That's why it is designated as sex chromosome. So this chromosome determines the sex of that particular organism. And the rest of the chromosomes, they were named as autosomes. Only this X chromosome that determines the sex of that organism. That's why it is called as sex chromosome. And rest of the chromosomes were named as autosomes. Got it? Then another example is grasshopper. Okay, and grasshopper is an example of exotype of sex determination. In this, males have only one X chromosome. In grasshopper, the males have only one X chromosome beside the autosome, whereas female have a pair of X chromosome. Okay, so in the case of grasshopper, autosome plus X chromosome. Okay, one X chromosome. They have only one X chromosome. Okay, but in male, this is in male. But in the case of female, they have a pair autosomes plus XX. That means a pair of X chromosome in grasshopper. Okay, you may get MCQs from this portion. Like in grasshopper, that is an example for sex determination, in which exotype of sex determination, in that the male have only one X chromosome with the autosome, but in female they have a pair of X chromosome, not single one. It is like a pair of X chromosome is present in female. Got it? So in large number of insects, the mechanism of sex determination is of X-type. That means all eggs bear an additional X chromosome besides the other chromosome or autosomes. And this was observed by scientists. So some of the sperm that bearing X chromosome, whereas some on the other side, some were not having that. So the eggs fertilized by sperm and do not have an X chromosome, that becomes male, and the egg fertilized by sperm that is having X chromosome, that becomes female. So in the male and female, the number of chromosomes are not equal. Okay. So in male and female, number of chromosomes are not equal, and it was called as sex chromosome, or they gave like a name like sex chromosome, and the rest of the chromosomes were named as autosomes. Okay, remaining chromosomes were known as autosomes. Only this chromosome is determining the sex of that organism. That's why it is called as sex chromosome. Okay, so the involvement of X chromosome in the determination of sex, that's why it is called as sex chromosome. And example of exotype of sex determination, in which the male have only one chromosome beside the autosomes, are seen in grasshopper. In which one? In grasshopper, you can see exotype of sex determination. In that, male have only one chromosome beside the autosome. And in female, they have a pair of X chromosome. Okay, they have a pair of X chromosome in female. Next, so all these observations, they led to the investigation of a number of species to understand the mechanism of sex determination. Okay, so these kind of observations that led to the investigation of number of species. They conducted experiment in more and more species to understand the mechanism of sex determination, how that sex is getting determined. They want to learn that, or they wanted to learn the mechanism of sex determination. So in a number of other insects and mammals, including man, XY type of sex determination is seen, where both male and female have same number of chromosomes. Okay, so usually up to this we learn the number of chromosomes. That is having variation in male and female. But here, the number of other insects and mammals. So there are number of other insects and mammals including man, that XY type of sex determination is C, where both male and female have same number of chromosomes. Okay, so in XY type of sex determination, we can see that both male and female both have same number of chromosomes. And among the male and X chromosome is present. Okay, in male an X chromosome is present, but the counterpart is distinctly smaller and called the Y chromosome. Okay, so among that chromosomes, in a male the X chromosome is present, but the counterpart is distinctly smaller and they are called as Y chromosome. Then in female, in XY type, in female they have a pair of X chromosomes. Okay, it's not single. They have a pair of X chromosome in XY type. They have a pair of X chromosomes. Both male and female bear same number of autosomes. Okay, in human beings, how you can see, same number of chromosomes will be present. Number of chromosomes will be similar. The number of chromosomes will be similar in male and female, right? So the male and female bear same number of chromosomes, or autosomes will be same number. So the male have autosome plus XY. In the case of human being, autosome plus XY in human beings. XY in human. Okay. But the female, they have autosome plus XX. Okay. So in XY type, to understand that mechanism, they tried with number of species, and female have same number of chromosomes in a number of other insects like Drosophila and mammals including man, and in plant also. In which plant? Melandrium. In that plant also, man, Melandrium and Drosophila, and all this and all, they conducted their studies and they found out that male and female both are having same number of chromosomes. So Melandrium, that was the first plant in which chromosomal basis of sex determination was discovered. Okay, chromosomal basis of sex determination was discovered in Melandrium. And in males, the X chromosome is present, but the counterpart is distinctly smaller, and that is known as Y chromosome. Okay, that is the Y chromosome. Then next is, in human being and in Drosophila, the male have one X and one Y chromosome. How it will be? X, Y. This is the condition of male condition in human beings and in Drosophila. In human and Drosophila. In both X, Y, that male, human being in male, they have one X and one Y chromosome. And the female have a pair of X chromosome besides autosome. Pair of X chromosome. Like this it will be. Instead of this XY, that is in male. Okay. And XX, that is in female. Okay. And next is there are two types of sex determining mechanism. That is XO type and XY type. Okay, two types of sex determination or sex determining mechanism. The mechanism of sex determination, that is of two types, XO type and XY type. Okay, XO type and XY type. But in both cases, male produces two different types of gametes. Okay, male produces two different types of gametes. Either that is with or without X chromosome, and some gametes with X chromosome and some gametes with the Y chromosome. Some with X chromosome and some with Y chromosome. Okay. So such type of sex determination mechanism, that is designated to be the example of male heterogamety. What is that? Male heterogamety. So gametes with X chromosome and some with Y chromosome. So that is not in homozygous condition. It is in heterozygous X and Y condition. So such type of sex determination mechanism is called as male heterogamety. What is that male heterogamety? That male gamete that is having X chromosome, some are having X chromosome and some with Y chromosome. That heterozygous condition or heterogametic condition, that is called as heterogamety. The type of sex determination mechanism is designated to be the example of male heterogamety. And in some other organism, birds, a different mechanism of sex determination is observed. In birds, some different mechanism of sex determination only obtained or observed. But in this case, total number of chromosomes, that is same in both male and female. The total number of chromosomes is same in both male and female. But two different types of gametes in terms of sex determination or sex chromosome, they are produced by female. Two different types of gametes in terms of sex chromosome, they produce female. That means female heterogamety. So in order to have a distinction with the mechanism of sex determination described, the two different sex chromosomes of a female bird has been designated to be Z and W chromosome. Okay. Is it clear? I'm explaining with this PowerPoint because it will be easy for you to write notes also. So in order to have a distinction with the mechanism of sex determination described. See, we have already described one sex determination mechanism. So these two different sex chromosomes of female bird, that is mentioned as Z and W chromosomes, or they were mentioned as Z and W chromosomes. So in these organisms, in which one? Female bird that is having Z and W chromosome, but the male have a pair of Z chromosome besides the autosome. So besides autosomes, that male have a pair of Z chromosomes. Okay, male have a pair of Z chromosomes. So in birds they have Z and W chromosomes, and the organism, the female have Z and one Z and one W chromosome, whereas male have a pair of Z chromosomes besides the autosomes. Okay, other than autosomes, they have a pair of Z chromosomes. Then The sex determination mechanism in the case of human. We are going to learn sex determination in human beings. Okay. So sex determining mechanism in human, that is of XY type. It's not of XO type, it is XY type. Sex determination in human beings is of XY type, no doubt. And out of 23 pairs of chromosome present, 22 pairs are exactly same in both male and female. Okay. These are called as autosomes. Okay. Total number of chromosomes in human being is 46. Total number of chromosomes. That means 23 pairs. Right? In this 23 pairs, 22. All 22 pairs, they are similar in male and female. In male and female, 22 pairs are similar. Okay. These are called as autosomes. Then a pair of X chromosomes are present in the female. But the presence of an X and Y chromosome are that is determined or that determines the male or female characteristics. So pair of X chromosome are present in the female, whereas presence of X and Y chromosome on male, that determines the male characteristic. Okay. Autosomes plus XY. This is male condition. Autosomes plus X. This is the female condition. Okay. So during spermatogenesis among male, two types of gametes are produced. Chromosome besides the autosomes. Then here, in the case of human, females produce only one type of ovum with an X chromosome. Only one type of ovum is produced with by the female, and that is with an X chromosome. So there is an equal chance of fertilization of the ovum that with the X or Y chromosome. So there is a 50-50 chance of fertilization of the ovum with a sperm that carries X, and there is another 50% chance of carrying ovum with Y sperm. So when an ovum fertilizes with the sperm, that is carrying X chromosome, that is called as female, or that forms the female, that develops the female. So in the fertilization of ovum with Y chromosome, that also produces sperm resulting in the, or that results in the sperm that results in a male offspring. Okay, that results into a male offspring. So ovum with Y chromosome that forms the male offspring, and ovum with the X chromosome in the zygote that develops into a female. So it is an event that it is a genetic makeup of the sperm that determines the sex of the child. Here, the genetic makeup of the sperm that determines sex of the child. And it is also evident that in each progeny there is always 50% probability of either male or female child. Okay, so there is a chance of 50-50 male child or female child. Then in our society, women are blamed for giving birth to female children and that giving birth to a female baby that was treated as a crime now, right? People are saying that if a girl baby is born, they will say that, oh, we didn't get a baby boy. Always they'll say like that. Actually, always they are blaming women for the birth of the baby. But what determines the sex of the baby? It depends upon male sperm. If male sperm is with XY, that forms XX forms a female, and when it is joining X with Y, that forms again male. Okay. So the sex determination in honeybee, that's what we are going to learn now. In human beings, we learned that was XY type. In honeybee, the sex determination in honeybee is based on number of sets of chromosome an individual receives. Okay, number of sets of chromosome an individual receives. So an offspring which is formed from the union that develops as female or queen or worker. Okay, offsprings formed from the union of a sperm and an egg that forms queen or worker. Unfertilized egg. If an egg is unfertilized, that develops into male or drone. Actually, that is like parthenogenesis. That is called as that process is called as parthenogenesis. Okay. So unfertilized egg, when the union of sperm and egg that forms or that develops as a female queen or worker, that is the formation of offspring with the union of sperm and egg that develops as female, that forms the females. And the unfertilized egg, the egg which is unfertilized, that develops a male. Finally, it will become male. And how it will develop? Due to parthenogenesis. Then that means that makes half of the number of chromosomes than that of the female. Okay, so the female, they are diploid having 32 chromosomes, and male means they are haploid, they are having 16 chromosomes. Okay, so the female that is diploid and it is having 32 chromosomes, and males are haploid and they are having 16 chromosomes. So this is called as haplodiploid sex determination. What is this? Haplodiploid sex determination. And so haplodiploid sex determination and special characteristic features such as male. Those males produce sperm by mitosis. Okay, those males produce sperm by mitosis, and they don't have father, and this cannot have sons but have grandfather and can have grandson. Okay. And next we are going to learn about mutation. What is mutation? Mutation means in this the alteration of DNA sequence. That is the mutation is a phenomenon which results in the alteration of DNA sequence. Okay, that consequently result in the changes in the genotype and the phenotype of organism. Okay, so almost every time these result of this DNA sequence that changes in the genotype and then phenotype of organism. First, this alterations in the genetic condition that changes the genotype and then the phenotype of an organism. That organism's phenotype will get changed. So, in addition to recombination, a mutation is another phenomenon. And see, we have learned about recombination. Then mutation is another phenomenon that leads to variation in DNA. Okay. So mutation means that is the alteration of DNA sequence. What do you mean by mutation? The alteration of DNA sequence. And that results in the changes of genotype. Due to that mutation or that alteration in DNA sequence, that results in the change of the genotype itself and that phenotype of an organism. That also getting affected by this mutation. So in addition to recombination, other than recombination, mutation is also present, and that led to the variation in DNA. Okay, so the DNA sequence is getting altered. DNA sequence got altered, then the genotype and the phenotype. The genotype and the phenotype, they forms or they show some kind of changes. So addition to this recombination, mutation is another phenomenon that this mutation or recombination means in any combination you can work. And mutation means there is a change, whole change in that particular sequence. That is called as variation. All these are called as variation in DNA. And one DNA helix runs continuously from one end to another in each chromatid, that also in highly supercoiled form. How it will be? It is highly supercoiled. Okay. So DNA helix that runs continuously from one end to another end in each chromatid. In each chromatid, the DNA helix will be continuously running from one end to another end. And that also it is in supercoiled form. So here the loss or the deletion of a particular segment or the insertion or duplication of a segment of DNA that results in the alteration in chromosome. That results in the alteration of chromosome. So the genes that are known to be located on chromosome and alteration in chromosome result in abnormalities or aberrations or differences. Okay, these mutations that will see, we have already told that DNA helix that is running continuously from one end to another end of each chromatid, right? That also this DNA, they are highly coiled or super coiled. So there, if there's some mutations or there's some deletions happening, means deletion or addition. That means some kind of changes. Sometimes one portion or one part is getting deleted. Then also it is an alteration or change. That is called as mutation. Or sometimes insertion. Something is getting added to that. Or duplication of a segment of DNA. That results in alteration in chromosome. Okay. Then that results in alteration in chromosome. So these genes are called to be, or these genes are known to be located on chromosomes, and alteration in chromosome that results in aberrations, abnormalities or aberrations. So these genes are located on chromosome. There is a slight change or there is a deletion or addition, means there will be some kind of changes, and this finally will result in the alteration of that chromosomes. So that the final result will be some kind of abnormalities or aberrations. Then chromosomal aberrations are commonly observed in cancers. So mostly the chromosomal aberrations, or they are commonly observed in cancer cells, and mutation also arises due to the change in a single base pair of DNA. Okay, sometimes this kind of mutation that arises due to change in a single base pair of DNA. Only a single base pair of DNA is changed, and mutation to change the single base pair of DNA, that is called as point mutation. Then we can say an example for such mutation is sickle cell anemia. Sickle cell anemia. So the example of such mutation, that is sickle cell anemia. Sickle cell anemia is an example for such type of mutation, or addition or deletion or of a single base pair. That means a small change. There is a single base pair is getting changed from the DNA. That is called as point mutation. So what is point mutation? Only that single point is getting added or deleted, or there is a change only in that single point. So such type of mutation is called as point mutation. The example is sickle cell anemia. And deletion and insertion of base pairs of DNA, that is called as frame shift mutation. What is frame shift here? Point mutation and frame shift mutation. Frame shift means deletion and addition. Deletion and insertion of base pair of DNA, that causes frame shift mutation. Okay. So there are many chemical and physical factors that may be the, sometimes that may be the reason for the mutation or the change. So these are called as mutagens. What are mutagens? The chemicals or the physical factors. Physical factors that induces such kind of mutation. They are referred to as mutagens. Mutagens or physical factors that induce mutation. That is called as mutagens. Sometimes UV radiation can cause, not sometimes, UV radiation can cause definitely mutation in organisms. This is called as a mutagen. So that's all about today's class. Do you have any doubt? You want me to repeat any part? Deepa, is it clear?
Yes, ma'am.
Oh. Okay. If you have any doubt, you can ask me in the next class. Thank you.
Thank you, ma'am.